Hello, everyone, and welcome to today's DDN webinar. I'm Yunning Wang, Associate Science Editor for DDN, and I'll be moderating our discussion today. We have an exciting webinar planned for you today. Our speakers, Dr. Galit Meshulam-Simon, Dr. Nicholas McGlincy, and Dr. Ankur Sarkar, will introduce a novel cell-free method for producing linear DNA templates that are ready for in vitro transcription for producing high-quality mRNA. This approach eliminates many of the bottlenecks associated with traditional plasmid-based systems, enabling faster, more scalable, and more consistent mRNA synthesis. After the presentation, we'll host a live Q&A session. To submit your questions or comments, simply enter them into the Q&A chat box on the right side of your screen. We'll try to get to as many of these as possible. I'd like to take this opportunity to thank our webinar sponsor, Allergen. Allergen is advancing the frontiers of synthetic biology with a free cell-free DNA synthesis platform that delivers high-quality IVT Ready DNA in record time. Their innovative solutions are helping to streamline workflows in mRNA therapeutics, genetic engineering, and beyond. Our sponsor has provided us with some helpful handouts related to today's webinar. You can find and access these in our handout section, located on the right side of your screen. Also in the handout section, you will find your certificate of attendance for participating in today's live event. And with that, let me introduce our speakers. First, we have our speaker, Galit Meshulam-Simon. Dr. Meshulam-Simon is an Associate Director of Commercial Applications at Allergen. She earned her PhD in biotechnology from the Technion Israel Institute of Technology. She brings over 17 years of experience directing R&D teams across a range of fields, including synthetic biology, protein engineering, infectious disease diagnostics, and AI-driven enzyme discovery. Her work has focused on advancing innovation and fostering partnerships in synthetic biology, molecular diagnostics, and genetic medicine. And then we have our second speaker, Nicholas McGlincy. Dr. McGlincy is a senior scientist on the commercial applications team at Allergen. He earned his PhD in RNA molecular biology from the University of Cambridge. His work focuses on understanding the mechanisms of gene expression and applying that knowledge to advance the development of innovative biotechnologies. And then we have our third speaker, Dr. Ankur Sarkar. Dr. Sarkar is a senior scientist on the molecular biology R&D team at Allergen. He earned his PhD in chemical engineering from the University of Colorado Boulder and focuses on combining molecular biology, synthetic biology, and protein engineering to develop innovative technologies that support drug discovery and improve human health. Galit, Nicholas, and Ankur, we're so pleased to have you guys join us today, and we look forward to hearing your presentation. Let's just take a moment to make sure your slides are up. Great, please take it away.
Hello and welcome to the webinar. My name is Galit Meshulam-Simon and I'm Associate Director at Commercial Applications at Allergen. With me today, Nick McGlincy, the Senior Scientist in the Commercial Applications team, and Ankur Sarkar, Senior Scientist in the Molecular Biology R&D team. Thanks for joining us to learn more about Infinia IVT Ready Linear DNA, powered by our cell-free, high-accuracy Infinia technology. We will highlight how this technology enables rapid, flexible, and reliable production of long linear DNA templates that are ready for mRNA synthesis. We will talk about product features and share data from example applications. Shipped in as fast as 10 business days with an encoded poly-A tail, Infinia IVT Ready DNA accelerate development timelines and support innovation in mRNA therapeutics. A little bit about Allergen. So Allergen, based in the San Francisco Bay Area, was founded in 2017 by Matt Hill to address the growing need for scalable, versatile gene synthesis. Elegant cell-free technology overcomes the limitations of conventional gene synthesis platforms that rely on cumbersome cell-based methods. Our production platform is highly scalable and adaptable to meet the growing demands of gene synthesis in all fields of synthetic biology, cell and gene therapy, and mRNA vaccines, without significant investment in resources or infrastructure. Conventional gene synthesis uses a hierarchical assembly process, starting with short oligos that are stitched or assembled together to create larger subunits. This is done using methods like as Gibson or Golden Gate assembly. This process is repetitive, time-consuming, and error-prone, introducing sequence error at each step. So to isolate the correct sequences, the DNA is typically cloned into plasmid, transformed into bacteria, and colonies are screened and sequenced just to find the perfect sequence molecule. These steps will be typically followed by a large-scale culture and plasmid preparation to obtain sufficient yield. This multi-week workflow is labor-intensive, costly, and limited by issues like gene toxicity and cellular contaminants. Elegant patented cell-free technology overcomes the limitation of traditional cell-based methods. We synthesize near-clonal quality double-stranded linear DNA that is longer and with higher complexity than offered by conventional gene synthesis provider. All of our linear DNA is producing this technology and is shipped globally from our facilities in the San Francisco Bay Area, United States. We'll introduce the Infinia platform products. So the first, as we discussed, is the Infinia linear DNA. This product was launched early in 2023, providing researchers with double-stranded linear gene fragments that are long, up to 7 kb, with very low error rate, in average 1 to 70,000 per base. So it's highly accurate, NGS verified, and ships as fast as 6 to 8 business days. The next product is Infinia plasmid DNA. What we realized is that to streamline cloning, our customers were using Infinia Linear to reduce fragment assembly. As plasmids remain essential in many workflows, Infinia plasmid DNA was launched early this year in January to provide long, complex sequences in ready-to-use plasmid format. And our recent new product where we will focus this presentation on is the Infinia IVT Ready DNA. mRNA is a programmable therapeutic platform with applications in infectious diseases, cancers, and genetic disorder. Despite its rapid adoption, which was highlighted during the COVID-19 pandemic, scalable production remains limited by issues of speed, fidelity, and batch variability. In vitro transcription using T7 RNA polymerase is the standard for mRNA synthesis, but plasmid-derived template poses challenges, which we will discuss soon. To address those, we used our Infinia linear platform to rapidly generate high-quality linear IVT ready template with encoded poly A tail of lengths of 70 to 130 base pairs. Traditional mRNA development is typically plasmid-based and involved fragment assembly, cloning, sequencing, and large-scale plasmid preparation that often takes weeks. Additional steps like linearization or sometimes PCR to add a tail is typically needed to meet the IVT requirement. At Elegen, our goal is to let scientists focus on discovery, not on DNA preparation. So our IVT-ready DNA bypasses plasmid workflows, enabling faster, more flexible mRNA design and screening to accelerate research. A bit about the challenges of the conventional plasmid-based methods. So what researchers typically doing is to build their gene of interest and clone it into a backbone that already includes the desired poly A tail. Then they propagate, purify the plasmid, linearize the templates, and purify again. So this process is long, and beside that, it requires many steps of purification and cleanup. Additionally, propagating and maintaining plasmid with poly A tails is not trivial, especially as the tails are getting longer, and that typically requires the use of special E. coli strains and ongoing QC. Although all that, it can still end with recombination products and large tail variability. Another plasmid-based method involves cloning into plasmid and adding the poly-A tail later by PCR amplification or by ligation. This might be slightly faster, but still takes longer time. And additionally, when you add poly-A tail by PCR, this could result in producing errors and tail polydispersity. So this is where Infinia IVT Ready DNA is coming, and it actually enables researchers to cut out the plasmid-based workflows and to get a fast template which is high quality and ready to use, enabling flexibility, quality, and speed. A bit about the Infinia IVT Ready product. So it's being synthesized using our cell-free synthesis, which is basically eliminating all the cell-based cloning, propagation, and maintenance, and extra steps to remove cellular contamination such as endotoxin, genomic DNA, and antibiotic residues. It just does not exist here. As for the tail, we offer two options for the encoded poly-A tail. This could be continuous in length, varies from 70 to 130 base pairs, or we also offer two versions of segmented tails. That allows the researchers flexibility and a platform to optimize their sequences. The researchers can also design their UTRs and promoters. As part of the Infinia platform, we offer two levels of complexity, standard and high, and enable up to 5.5 kb length of the coding region, not including the tail. We also ensure high quality. All the shipped constructs are NGS sequence verified and are going through our robust QC process to ensure best performance. And it's fast. The construct can be shipped as fast as 10 business days. Overall, Infinia IVT Ready linear DNA enable researchers to get their templates ready for rapid in vitro and in vivo testing and to accelerate development cycles. At this point, I will invite Ankur to talk in the next slide about the production process of Infinia IVT Ready DNA, and he will demonstrate the process robustness and the quality of the.
All right, thank you, Galit. So in preparation for launching IVT Ready DNA, we really wanted to extensively test our process and make sure that we could robustly deliver the product that our customers needed. So to that end, we tested 319 distinct IVT Ready DNA sequences. 91 of these were synthetic constructs based on Infinia orders we had fulfilled previously. And we modified these to evaluate how different sequence complexities would affect build success of the IVT Ready DNA product. In addition to that, we had 228 sequences submitted by beta test customers that we also tested to see how robust our process was to the types of constructs customers are interested in. This panel spanned the range of complexities that is shown in the table on the right. So we looked at sequences ranging from 1 to 5.5 kb, global GC content from 36 to 64%, local GC content within 100 base pair windows spanning from as low as 13% up to 80%, as well as large secondary structure forming features such as repeats and hairpins. The first thing that we confirmed was that we could reliably synthesize across the different lengths that we offer, as well as across different sequence complexities. So this table shows for either below 2 kb, 2 to 4 kb, or 4 to 5.5 kb constructs, how successful we were in synthesizing our constructs. So for example, less than 2 kb standard complexity constructs, we were able to successfully build 98% of the 188 we tested. And across all the ranges of lengths and complexities, we are at about 90% or higher success rate. And this success is based, as Galit mentioned, on both our NGS QC, similar to Infinia, every sample is sequence verified, as well as meeting the minimum yield spec that we promised. One thing that we're very excited about with this product is the ability to offer customers to add lots of different poly-A tails to their constructs. We offer as low as A70 all the way up to A130 poly A tails, as well as 2 segmented tails containing the well-known 10 base pair UGC linker. And so we confirmed that with constructs that were both standard and high complexity across a wide range of the tails we offer, we're again able to successfully build 90% or more of the constructs that we attempted. In addition to offering many different tails, we're really excited to offer customers the opportunity to test many different tails with a given construct. So to that end, we investigated how the same IVT-ready DNA construct could be built successfully with different poly A tails. So an IVT-ready DNA construct would span, for example, the promoter through the end of the 3' UTR in this figure that's shown on the slide. And we tested a number of constructs that were appended with different poly A tails. So, for example, the first row in the table shown on the right shows that we tested 7 constructs with both the A70 and the A130 tail and successfully built 6 of those with both of those tails. And you can see for all the different combinations we tested, we were making most, if not all, of the constructs with multiple tails successfully. With the ability to add different poly A tails, it's obviously very important to confirm that our process is actually producing molecules with tails of the expected length. So to evaluate that, we use long-read PacBio sequencing to look at tail length. And what I'm showing in the plot here is 82 constructs built with an A120 tail, and we're looking just at the distribution of poly A tail lengths in the tails of those molecules. So you can see that over 90% of the tailed molecules are within 85% of the target length, as shown by the gray shaded box there. Later in the talk, you'll see some data for additional tail lengths, as well as how our product compares favorably to traditional plasmids with respect to poly A tail length and dispersity. Finally, we wanted to confirm that we could deliver the promised yield to our customers across lengths and complexities. So in the plot shown here, the dashed line at 10 micrograms is the current promised yield that we offer for Infinia IVT Ready DNA. And you can see that across lengths from 1 all the way up to 5.5 kb, as well as across IGC and secondary structure forming features, we're able to consistently deliver yields that meet that spec. So at this point, I'm going to turn it over to Nick to tell you a little bit about the functional performance of Infinia IVT Ready DNA as compared to plasmids.
Hello, everyone. Today I'm going to be talking about, as Ankur said, the functional performance of Infinia IVT Ready DNA as compared to plasmid templates. So to assess the functional performance, we designed 4 constructs. mCherry or Firefly luciferase open reading frames driven by a T7 promoter with the same 5' and 3' UTRs, and then each with either a 90 base pair or a 130 base pair poly A tail. All 4 constructs were classified as high complexity. In the case of mCherry, this was due to the 5' UTR used, while for FLOOK, the UTR, the ORF itself, was high complexity. These constructs were then built both as Infinia IVT Ready DNA and ordered as plasmids from an external supplier A. As you can see from the table, Elgin achieved a faster, more consistent turnaround time than supplier A, delivering all constructs in 12 business days. In contrast, we ordered our plasmids from supplier A as a premium service with an expected turnaround time of 18 to 34 business days, but then experienced a turnaround time of 15 to 52 business days. What is more, the 130 base pair poly A tail is not a standard offering for supplier A, and they were unable to build the FLOOK construct with a 130 base pair poly A tail, instead delivering a 123 base pair poly A tail that was confirmed by Sanger sequencing. This experience underscores the improved flexibility and reliability in poly A tail length construction that has been achieved with Infinia IVT Ready DNA. The variability in poly A tail length for these DNA templates was further examined by PacBio long-read sequencing. This graph illustrates the poly A tail sequence length distribution for all 8 constructs. So on the x-axis, you can see that we have the poly A sequence length in base pairs. On the y-axis, we have the percent of reads. The vertical facets are the open reading frame encoded by each of the templates, either mCherry or FLOOK. And then the horizontal facets reflect the designed poly-A tail length, so 190 or 130, though as I mentioned earlier, we're only expecting 123 base pairs from the F-look construct. In each facet, there are two traces. The blue-green is the IVT Ready DNA, and the gray is the plasmid-derived DNA. So what you can observe from this data is that the centering of the distribution around the expected size of the poly-A tail, which is the dashed line in each case, and the tight dispersity of the poly-A tail are comparable between the two template types and within our 15% spec. However, the plasmid templates, the gray lines, have a higher proportion of truncated poly-A tails, which is particularly evident for the 130 base pair poly-A constructs, as I pointed out by the red arrow here. This can be observed through the flat secondary peak in the gray trace here, and the lower proportion of reads that have a poly-A tail within 15% of the target length, as we outlined in these 2 points here. Okay. To test the functional performance of these templates, 500 nanograms of Affinia IVT Ready DNA or linearized plasmid was then used as a template for IVT reactions using Thermo's Megascript T7 transcription kit. The RNA yields, as determined by nanodrop, are illustrated in the bar chart on the left-hand side of this slide, where the height of the bar represents the yield in micrograms of RNA, and the error bars is the standard error of that mean. In each group, the mean is the mean of 3 to 6 replicates. So moving from left to right, we have M-cherry with a 90 base pair poly-A tail, M-cherry with 130 base pair poly-A tail, F-look with a 90 base pair poly-A tail, and finally F-look with a designed 130 base pair poly-A tail. The gray bars represent the plasmid-derived DNA, and the blue-green bars represent the IVT Ready DNA. We observed that an equivalent input mass of Affinia IVT Ready DNA resulted in an approximately 1.5-fold increase in RNA yield compared to the linearized plasmid template. We also observed the expected increase in RNA yield for the longer F-look template. This observation of increased RNA yield also held when we titrated the input DNA mass of the M-cherry 90 base pair poly-A tail for both template types. Here in this dot plot on the left-hand side, each dot represents a single IVT reaction, and the lines represent the low S regression fit of those dots. The blue line is the IVT Ready DNA, and the gray line is the corresponding plasmid-derived DNA. Along the x-axis is the input DNA mass in nanograms, and on the y-axis is the total RNA yield in micrograms. We believe that this effect is driven by the higher molarity of a given mass of Affinia IVT Ready DNA, where the template only contains the sequences required for IVT and none of the sequences that a plasmid must carry in order to be propagated in the bacterium, such as an origin replication, resistant markers, etc. Moving on to qualities of the RNA, further qualities of the RNA, the production of a single RNA species of the expected size is a critical quality attribute for many of the applications of in vitro transcribed RNA, especially mRNA therapeutics and vaccines. We assayed this by running appropriately diluted RNA samples on an Agilent 2100 bioanalyzer using the RNA 6000 pico kit according to the manufacturer's instructions. Along the x-axis, we see the size of the RNA in nucleotides. Along the y-axis, we see the fluorescence data provided by the bioanalyzer. In the vertical facets, we have the length of the poly-A tail, either A90 or A130 designed. And then on the horizontal facets, on the top, we have the plasmid-derived RNA that has come from a plasmid-derived template. And then on the bottom facet, we have the RNA that's come from the IVT Ready template. So as you can see, the size and position of the main peaks are consistent with the expected RNA sizes, which is the dashed vertical line, plus minus 15%, which is the grayed out box, and comparable between template type. Furthermore, each main peak represents greater than 85% of the total area under the curve for each trace. The poly-A tail of an mRNA is a vitally important functional element. It both promotes transcript stability and translational initiation. As such, the length distribution of the mRNA poly-A tail is a further critical quality attribute for many mRNA therapeutics. We measured poly-A tail length distribution of our mRNAs using a mass spec-based assay, complementary approach that can avoid biases that can be introduced by library preparation and high-throughput sequencing. RNase T1 is a nuclease that specifically cuts after Gs in RNA. Thus, the major digestion product of an mRNA is expected to be the poly-A tail. The length distribution of the poly-A tail can then be accurately determined by examining the size distribution of the digestion products by an LC-MS method. The results of this data can be seen on the plot on the left-hand side. Along the x-axis, we have the mass in daltons of the resultant species, whereas on the y-axis, we have the abundance, here measured as the percent of the most abundant species. The vertical facets are the open reading frame, which is encoded by the mRNA, either F-look or mCherry. And then on the horizontal facets is the template type that the RNA was derived from, either plasmid-derived or IVT Ready DNA. As you can see, for both template types, the observed mRNA poly-A tail distribution is closely aligned with the expected length. The rightward shift of the length distribution is the result of a well-characterized phenomenon termed transcriptional slippage, where T7 RNA polymerase slips while transcribing homopolymer stretches, and this results in an elongation of the poly-A tail. Finally, the ultimate functional test of our RNA is whether it produces functional protein. To assay this, we made capped mRNA for our F-look encoding constructs using NEB's high-scribe CleanCap AG kit, and used an equal mass of RNA to generate protein using Thermo's one-step human in vitro translation kit. F-look protein expression was then assayed by measuring the luciferase activity of the protein extract using Beers's glow assay. The results of this assay are seen in the plot on the left-hand side. Along the x-axis, we have the log 10 of the luminescence, which is represented as the mean plus minus the standard error of that mean. And what you can see is here we have the F-look constructs with an A130 tail, the gray bar being that derived from the plasmid template, and the blue bar being that derived from the IVT Ready template. And then next down, we have the same results, but for the A90 constructs. We then have the negative control, which was an empty reaction that was not programmed with RNA. And then finally, we have the positive control, which is an equivalent mass of CleanCap Firefly luciferase mRNA purchased from TriLink. What you can see from the data is that the observed luciferase activity is comparable both between the different template types and between the different poly-A tail lengths. In conclusion, over the course of this presentation, we have talked about the characteristics of Infinia IVT Ready DNA and how it provides a high-quality, rapid, and flexible platform for generating mRNA. We have demonstrated our robust production process based on the Infinia platform that allows us to span a broad sequence design space while maintaining high quality. Furthermore, we have examined functional qualities of the mRNA generated from both IVT Ready DNA and equivalent plasmid templates. We found that the IVT Ready DNA produces RNA that is comparable or superior in all the measured endpoints. Thank you very much for listening. If you would like to learn more, please reach out to our customer support email or visit our website, and look out for further published material in the coming weeks.
Thank you for that great presentation, Galit, Ankur, and Nick. And let's move on to the Q&A session. We have a few great questions coming through the chat already, and let's dive into them. The first one is, are there any specific design considerations when ordering the sequences? Any of you like to take that one?
Yeah, sure. I'm happy to take that one. So yeah, there are a few different design considerations, and importantly, you don't have to remember what I say. You can also check on our website to confirm that your sequence is being designed correctly. So similar to Infinia DNA, we offer standard and high complexity orders. One thing to note for IVT Ready DNA is we offer sequence length from 1 to 5.5 kb, excluding the poly-A tail. So if your sequence is shorter than that, be sure to include some random sequence at the 5' end. A couple of other features to think about. So obviously, consult with the IVT kit or manufacturer that you're working with, and make sure to include a promoter and the necessary UTRs, and if you need it, a sequence for capping, just to make sure that the RNA is going to work in your downstream application. As we mentioned in the talk, We offer a set of tails spanning from A70 all the way up to A130, as well as two segmented tails that include the well-known 10 base pair UGC linker. And if you have sort of design considerations that extend beyond any of that, please reach out to anyone on the customer service team, and we're happy to address the specific constraints of your design.
Thank you. That's a great overview of the design factors researchers should keep in mind. The next question is, how should I use Infinia IVT Ready DNA in my applications? Nick, would you like to take that one?
Certainly. So IVT Ready DNA is ready to use. All you need to do is to respend the lyophilized DNA template in molecular biology grade water or buffer and use it directly in your IVT reaction. There's no need to amplify the sequence or to add a poly-A tail or to linearize as you would have to with plasmid. In terms of the amount to start with, we found that a good starting point would be the same linear template mass as you would use for plasmid-based templates and optimize from there. As you have seen in our presentation, our experience has demonstrated that in many cases, depending on the kit and the gene transcribed, one can often use less linear template from IVT Ready DNA to achieve the same RNA mass due to its higher molarity per unit mass. So you can get rapidly from designs to results in as little as 10 business days.
Sounds great. Thanks, Nick. The next question is, how do you QC the product? Would anyone be able to answer this one?
Sure. Yeah, I can take that one. So again, similar to our Infinia DNA product, everything that we ship that's IVT Ready DNA is verified for yield, purity, and accuracy with qubit fluorescent measurements for yield and NGS verification for purity and accuracy. Our data and external data has shown that this process yields high-quality templates that perform equal to or better than plasmids. And as we showed in the presentation today, we've also confirmed that the QC product that we build is able to deliver tails of the desired length and polydispersity.
Thanks for sharing that. So building on that, can you comment on whether the DNA template is free from any undesirable contaminants?
Yes, I can address that question. Thank you. So as Infinia IVT Ready DNA is generated cell-free, it is largely free from the contaminants that plague preparations of plasmids. So things such as endotoxin, bacterial genomic DNA, and antibiotic residues.
That's good to know. Thank you, Nick. Will the IVT reaction yield sufficient quantities of mRNA? Galit, would you like to answer?
Yeah, that's a great question because we know that maximizing the mRNA yield is a primary concern for our experimental, for the researchers and experimental efficiencies. So what we see is the Infinia IVT Ready consistently produces the same yield or even higher when compared to linearized plasmid. And as we have shown that, we saw that in our own experiments, we also hear that from early access clients. And many times it will be even higher than this when you compared with plasmid using the same mass input, probably due to the high molarity of the Infinia IVT Ready templates.
That's very reassuring. Thank you, Galit. The next question is, can you comment on the mRNA integrity and the poly-A tail dispersity? Ankur, would you like to answer that one?
Yeah, sure. Happy to. So mRNA integrity is obviously very important. You need your full-length intact transcripts to get the function that you want. And it's known in the literature that IVT protocol conditions and reaction components have a big impact on this, including downstream purification steps. However, we apply, as I mentioned earlier, pretty strict QC to our high-quality DNA templates. And as a result, that strictly QC'd material in both our experiments and external data is able to consistently produce mRNA with comparable or higher integrity than linearized plasmids. And as you saw a little bit in this talk, we're also able to deliver comparable or better tail polydispersity compared to linearized plasmids.
Sounds great. Thank you. Will the mRNA produced be functionally active and potent in biological system?
I can take this one. Yeah, this is a serious concern. This is often the ultimate test for any synthesized RNA. So as you saw, we've benchmarked mRNA from Affinia IVT Ready DNA templates against plasmid-derived templates for yield, purity, integrity, and potency. Affinia showed comparable or superior performance across all metrics. Our product has also been demonstrated by collaborators to be functionally active, achieving comparable or even slightly higher on-target editing efficiencies in ex vivo gene editing studies when compared to plasmid-derived templates.
That's great to hear. Thanks, Nick. The next question is about if the process can be easily scaled up for larger research or therapeutic needs.
Yeah, I can take this one. So definitely scalability is a main consideration, especially when progressing from discovery to development and production. So current order can be shipped at 10 microgram scale. And if you need more, please reach out to your Elogen representative. We have options and we'll be happy to assist you.
Wonderful. Sounds like the platform is quite flexible. The next question, do you ship outside the US?
Yes, we do. So we make all our products in San Francisco, Bay Area, in the United States, but we ship globally. Overall, Infinia products are shipped dry at ambient temperature, dry down in a 96 well microplate with 1 sequence per well.
That's good to know. So we have time for one last question. To close things off, Galit, could you summarize the key advantages of the Infinia IVT Ready linear templates for us?
Yes, I can do that, and happy to hopefully what will summarize this presentation. So there are many advantages to this product. I'll start with the fast turnaround time. So depending on the sequence complexity, but sequences could be shipped as fast as 10 business days, and they are NGS verified. So this is really compared with much longer time that takes to plasmid-based templates. The second important point is flexibility. I hope that we were able to show you that we can have many sequences that are passing robustly and reproducibly during our platform. So sequence can be of different lengths or different complexity. The poly-A tail can be continuous or segmented, different poly-A tail lengths. So all this is to enable our researchers really to optimize and screen and do that as fast as they can. The product is ready to use. You don't need to amplify, you don't need to poly-A tail, you don't need to linearize, you just take it and you resuspend it and just go directly into a IVT reaction. Another main advantage is really being a cell-free product. So by really cutting out plasmid out of the workflow, there's no need to worry about bacterial strain on how to propagate and maintain the poly-A tail and the integrity. You basically get a product which is free of all that. It's free of leftovers of bacterial cells or genomes, and you can just go ahead and use it. So I think that overall, Infinia IVT Ready DNA templates are high quality. They are producing mRNA, which is comparable or even better to plasmid-based templates, and their fast turnaround time shortens the development time and accelerating mRNA discovery and optimization. So yes, please follow us. There will be more published material coming soon. Reach out in any question. Our team here is happy to support you, and hopefully you will enjoy the product. Thank you for listening.
Fantastic. Thank you for that excellent summary. And that brings to the end of the session today. If you have any further questions, please feel free to reach out to our speakers directly. Their email addresses are now displayed on the screen. Today's webinar will be archived on the DDN website, and you will receive an email notifying you when it's available on demand. On behalf of DDN, I'd like to thank our speakers, Galit, Nick, and Ankur, as well as our sponsor, Elogen. And of course, thank you for tuning in. Have a great rest of your day. Goodbye.